Good day, everyone. Jake and I will report our case study entitled Parasitic Protozoans in Farm Fishes and Crustaceans. Protozoans are also microscopic organisms with specialized structure for movement, food gathering, and attachment. They can be external or internal parasites. They can multiply on or within their host. Parasitic infections often give an indication of the quality of the water, since parasites generally increase in abundance and diversity in more polluted water. Objectives to identify the parasitic protozoans on farm fishes and crustaceans, to discuss the practical key of protozoans on fresh species, to discuss on how to control the future diseases caused by protozoans.
So good day everyone. So today I'm going to discuss the types of protozoans, namely amoeba, dinoflagellates, chenoplastid flagellates, diplomatoid flagellates, apicomplexans, microsporidians, and xylates. So first is the amoeba. Amoebas are eukaryotes. They're single cells, like those of other eukaryotes. They possess certain characteristic features, such as their cytoplasm and cellular contents are enclosed within a cell membrane, and their DNA is packaged into a central cellular compartment called the nucleus. One species of this is the Neoparamoeba purulans, a parasite causing amoebic gill disease in Atlantic salmon in marine fish farms, and it is one of the best documented gill disease in salmon farming, eliciting high morbidity and some mortality. So next is the dinoflagellates. So dinoflagellates are mostly marine plankton. They are also in freshwater habitats. Their populations vary with sea surface temperatures, salinity, and depth. One species of this is the Amolodinium ocellatum. This species has an ectoparasitic lifestyle and has been considered a pest in marine aquaculture for decades due to its association with severe morbidity and mortality. Next is the chenoplastid flagellates. So this type of flagellate as a species named Ichthyobodonecator. This flagellate is found both in freshwater and marine facilities. There is debate whether more than one species is involved, and recent studies have provided molecular clues to differentiation of different strains and species within the genus Ichthyobodo. Next is the diplomonadid flagellates. One species of this is named Spironucleus salmonis. It was originally described as Hexametas salmonis. It is a common intestinal parasite in farmed rainbow trout, eliciting intestinal dysfunction and weight loss probably due to malabsorption. Next is the apicomplexans. A number of apicomplexan species are common in wild fish from both freshwater and marine habitats. However, reports on apicomplexan infections in maricultured fish are few. When compared parasites infections in wild and maricultured COD in Norway, they detected Gosia spraguei as the only one species within this group and occurring only in wild fish. However, farms in northwest Spain were diagnosed with Cryptosporidium scophthalmi. Next is the microsporidians. A range of microsporidians has been detected in various maricultured fishes. One of these is the Loma salmonae in salmonids, but also Pleistopora angularium in Japanese eels or the Anguilla japonica, and Enterocytozoon spp in Atlantic halibut hypoglossus, and have been associated with pathological reactions in farm fishes.
External protozoans. First is the Ichthyopteris longifilus. Is the newest protozoan parasite penetrate the epithelial tissue of the skin and gills of the fish. It is perhaps and mostly readily recognized protozoan fish parasite. It is commonly referred to as itch or white spot disease. The The large parasite cause multiple small raised white lesions that develop as a result of the parasite residing in the skin, fins, gill tissue of the host. The Tetrahypermia piriformis. It is another fluid that occasionally causes problems by invading the deeper tissues of fish. In Tetrahymena piriformis, this parasite causes death or disease in a number of species of tropical aquarium fishes, especially guppies, neon tetras, and mollies. This multi-shaped organism penetrate the epithelial tissue and continue migrating along the facial planes of the underlying muscles to invade many of the internal organs. Third is the trichodyna. These parasites of freshwater brackish and marine species are readily identified by their internal circular ventricular ring that has both an inward and outward facing with of tooth-like projections. Those are parasites that are generally most pathogenic for they cause significant tissue irritation, hyperplasia of epithelial and respiratory problems. Fourth is the Chicodonelia species. It is the dorsoventrally flattened oval protozoans that has cilia located its distinct bands along the surface of the body. This freshwater parasite causes irritation of the gill's fins by its feeding activity while resulting in hyperplasia and fusion of gill laminae and hyperplasia fin tissue. Internal protozoan parasites. First is the Spirinucleus species and Hexametine species. They are the small flagellate parasites that are frequently found in the lumen of the intestinal tract of freshwater and marine tropical fishes. Mild infestations of the intestinal tract are generally asymptomatic, while heavy infestations can be pathogenic, causing necrosis and slowing of the intestinal epithelium. The resulting clinical signs include inappetence and thriftiness, mucoid or pale skin, poor condition, evacuation, and death. Second, the microsporidium. Microsporidium is a parasite produces spore-filled yeast within almost any tissue of many freshwater and marine species. As the cyst gets larger, infected muscle tissue becomes displaced and turns white in color. Blastopora hypesobriconis causes a syndrome called neon tetra disease in Severianus, cichlids, and cyprinid.
So next is the practical key for the determination of fish protozoa in fresh material. So protozoa can be detected as microscopical whitish aggregation from tiny dots to cyst-like structures of several millimeters or even centimeters in size, on the skin or gills or on the internal organs or in their tissues. Determination is only possible by the use of compound microscope. Protozoa is visible as tiny dots on the body surface and gills. Under the microscope, the dot proves to be the one of the several large, up to 1 millimeter, slowly rotating cells uniformly covered with synchronously beating cilia next to large ones. There may be small ones of different size. Their cytoplasm is full of granules and contain a large central macronucleus silice of the genera Ichthyopreteus, Thyreus, of Cryptocaryon. The protozoa are spherical or ellipsoidal bodies of about 10 to 20 size, each containing 4 ellipsoidal bodies, each of which contains 2 slender cells. Whitish nodules within the body organs are not sharply delimited Caucasian oocysts.
Disease control. First is the chemical control. Many diseases in fish are caused by ectoparasitic protozoans invading the skin, fins, and gills. These organisms fly free, living in life cycle stages, which make it possible to control the infections through water disinfection by use of substances such as copper sulfate and formalin. Due to the environmentally problematic effects of the compounds, recent studies have recommended alternative uses of oxidizers such as hydrogen peroxide and related compounds. Second, medical control. Several drugs which anti-protozoans effect has been developed and area viable for treatment of humans and domestic animals. However, also experimental work has demonstrated marked activity against fish protozoans. Their use within aquaculture enterprises is limited due to lack of licensing to legislative restriction. Third, management. Strict hygienic measures, quarantine practices, compulsory use of certified disease-free fish following production sites, and elimination of infected parasites. Staged by various filtration techniques are practices which should be used in any production system. Fourth, breeding of innately resistant fish. Mechanism involves the responsible for innate resistance of specified stocks, and species of fish towards pathogens have been fully elucidated. However, a range of examples is available. A well-documented example is the innate resistance of several Baltic salmon strains against the ectoparasitic monogenin, the G. salariensis. Fifth, immunoprophylaxis. The potential and the capacity of the cell's immune system can be appreciated through recent discoveries which has suggested that immunoprophylactic measures may be significant ways to secure fish health. Some strategies in the field are ways of feeding fish with compounds often immune which chemicals stimulating innate immune parameters. Other strategies apply specific immunization of the production animals against pathogens. 6. The immunostimulants, a series of structural molecules from various sources of bacteria, yeast, fungi, and lichens, along or together with the synthetic products. The immunostimulants possess pathogens-associated molecular patterns which are able to stimulate different pathogens recognition receptors in host fishes and thereby activate innate immune reactions. 7. The vaccination. Disease control in aquaculture enterprise is ideally based on integrated system, applying high hygienic standards. This can be achieved through filtration of water, securing optimal by chemical water parameters by breeding for species strains and feeding at an optimal level. However, additional control methods must be considered in order to avoid medication. Vaccination is an immunoprophylactic measure which can provide the fish with high resistance against infection.
So next is the parasitic protozoans on farm fish and crustaceans. The species black tiger shrimp or Penaeus monodon serve as the host for a wide range of protozoan parasites and commensals. Protozoan parasites and commensals occur both inside and outside the host body. Peritrichus ciliates like Zootamnium sp and Voricella sp are ectocommensals, and Microsporidia and Gregorines are endocommensals. The ectosymbionts Periticus ciliates remain attached to the gills and limbs. A possible relationship between the peritrich ciliate, zootomium sp, and mortality of host following stress was discussed previously. The ectosymbionts Peritricus gregarine, belonging to the phylum Apicomplexa class Protozoa, order Eugranidae, are all pathogenic, although they cause some local damage and occlude passage in the inhibited organ with serious results. They reside in the gut of the decopods crustaceans and may lead to reduced absorption of food or occasional intestinal blockage and possibly mortality of their host. Two types of Gregorians, Nematopsis sp and Cephalobus sp, are encountered in penid shrimps. Invasion with Gregarianids vary with temperature. The infection rate with Gregarianids is high in summer than winter. Microsporidians belonging to the phylum Microspora class Microsporia order Microsporida are another type of obligate intercellular parasites of shrimps and prawns. They can infect a wide range of invertebrate and vertebrate taxa, but almost half of their described species have insects as their host. They may infect gut, hepatopancreas, muscle, reproductive tissue, and nervous tissue. There is no seasonal trend in the prevalence of the parasite in crustaceans.
Therefore, protozoa can cause diseases to the aquatic organisms that may affect their growth. It has different practices to prevent protozoans appears in those species in the aquatic organisms that we could practice to prevent and must know the basics on how we could treat the protozoa. In addition, parasitic protozoans can avoid and even do necessary things in preventing these diseases to our commodities, just like proper management control, and it must be monitored once in a while. Thank you for listening to our report. Advance Merry Christmas and Happy New Year.